My name is Britton Goodale, and I am a postdoctoral fellow studying arsenic as part of the Superfund Research Program at Dartmouth. I've been studying arsenic for almost 3 years. When people we know or people we love are sick with diseases, we often look for reasons why. Since I was a child, I really wanted to be able to really know whether a chemical would cause something like cancer or other diseases. And so that's why I became a toxicologist. The goal of our research in the Stanton lab is to understand how arsenic decreases the ability of organisms to respond to stressors in their environment. It's in the bedrock, and it gets into drinking water. It also gets into our food. So we're not able to completely eliminate arsenic exposure. And that's why we need to work together across disciplines to figure out ways that we can reduce exposure, and that includes filtering water, different methods of growing crops so that less arsenic gets into our food, and we need to figure out ways to help people to test their water and treat their water. Arsenic is a major environmental contaminant of global concern. It affects hundreds of millions of people, and long-term exposure to arsenic through drinking water is associated with many different diseases, and this includes cancer, both lung cancer and bladder cancer, and also diabetes and lung disease, which is what we study. We grow human lung cells in the lab, and we're able to keep them alive by feeding them a liquid media. So epithelial cells line the surface of the lung. It's a thin barrier because gas exchange needs to happen as you breathe. So if you think about it, it's a really great place for a pathogen to invade and get access to the body. So in my study, we are exposing the epithelial cells to arsenic, and then we add Pseudomonas aeruginosa, which is an opportunistic lung pathogen. And what we find is that oftentimes a low dose of arsenic, it won't kill the cell, or we might not even be able to detect that anything's wrong. The only time that we can see that something is not right in the cell is when we add that additional stressor. We measure levels of RNA in cells that are not exposed to arsenic and compare them to cells that are exposed to arsenic to essentially see whether genes are turned on or off. We use different kinds of software on our computers to look at the data and to make sense of all of these thousands of genes that we look at a time, and what this is called is the field of bioinformatics. That's a real key part of what I do in my research. When we collect data and then go back to our desks to analyze it, when you have a new data set, being able to see things that nobody else has seen before feels kind of like being an explorer, and that's really exciting.